So right now, I'm really excited to introduce a live presentation via Zoom with Dr. Angela Slitt. So Dr. Angela Slitt is a professor of biomedical and pharmaceutical sciences in the College of Pharmacy at the University of Rhode Island. She has more than 20 years of experience as a toxicologist, specializing in mechanisms of liver injury and biliary excretion transport, with numerous full-length publications in the area of toxicology. So without further ado, Dr. Angela Slitt.
Hi, can everyone hear me? Hello?
Yes, we can hear you.
Okay, great. Sorry, I couldn't hear the speaker. So good morning, everybody. Thank you for the invitation to present our research regarding PFAS. While our laboratory is specifically funded to study PFAS, obviously the implications that it has for components of AFFF and the health and wellness of firefighters is clearly relevant. So thank you to the organizers for inviting me to speak about our work today. My name is Angela Slitt, and I'm a professor in biomedical and pharmaceutical sciences at the University of Rhode Island. At the University of Rhode Island, we are funded jointly through the Harvard Steep Superfund Center to utilize interdisciplinary science to address the human health concerns around PFAS. And I just want to give you a big picture of kind of how our laboratory supports the Superfund's efforts in understanding the health effects of PFAS and how it all fits in. And so as firefighters, I know some of you out there are working hard fighting fires. I just wanted to take a minute to kind of briefly explain to you how our science works and at the University of Rhode Island. So we have different projects that feed us information. And so for example, we are working with scientists who measure the levels of PFAS that are present in Cape Cod well water and different Cape Cod communities that are impacted through AFFF contamination. And those particular collaborators and scientists give us information about the types of PFAS that they're detecting, the amounts, and the relative combinations of all of them put together. And so that impacts our testing. So that gives us an idea of just not what PFAS are that we know are there, but maybe ones that are new and able to be measured through new methods. We also work with collaborators who are detecting PFAS in the bloods of Faroese mothers and children. And while these individuals are in Denmark, they're a very controlled population. And so because of that, they can give us information about exposure and then how that exposure relates to human health. So this group tells us about what PFAS we should be studying in our cell-based models that are being detectable in human blood, human umbilical serum, and breast milk. And then lastly, some of our collaborators measure what's out in the environment, whether it's in water, in this case, it could be in the livers of marine birds, and can say, okay, these are what we're detecting out in our sentinels across the environment. Can you test these in the laboratory and help us understand whether or not their behaviors are similar or different to the PFAS that we know about, such as PFOA and PFAS? And so here our laboratory comes along, and what we do, for today, I'm going to talk to you about our work in human hepatocytes, is we take those hepatocytes and we plate them and assays that we can kind of get a lot of information about those PFAS relatively quickly, and test these new emerging PFAS against ones that we already know, and then get an outcome related to disease. In our case, we're interested in understanding whether or not these can be involved in aspects of liver disease. So specifically, we're interested in how PFAS affect the liver. And we know that the liver is the tissue where there's that, at least for the legacy PFAS and some of the PFAS replacements, the liver is often a site of accumulation and distribution for PFAS. And we know that some livers can have relatively high concentrations and higher concentrations even than the serum, depending on the PFAS. So we know liver is a target organ. And in particular, for the liver, in human studies, we do see associations between PFAS and PFOA with regard to markers of liver injury. So when you go to the doctor and the doctor measures liver injury, looking at, say, elevations in serum liver enzymes, there are many studies that show an association between PFAS and PFOA and elevated liver markers. We also know that PFAS affect liver size in rodents. This tends to be a rodent-specific phenomena, but we do know that it causes what's called hepatomegaly, and that's when the liver gets bigger. Now, this particular observation is somewhat debated within the literature in whether or not it's relevant to humans because of species-specific differences. However, when PFAS are evaluated within the regulatory world, what one consideration is what's called the Hall criteria. And whether that's, and really what it says is that you have to consider increased risk for cancer if that particular PFAS causes hepatomegaly, so liver enlargement, and also cytotoxicity. It increases those liver enzymes. So there are PFAS that meet this Hall criteria, such as PFOS and PFOA. Now, in addition to that, there are some rodent and cell studies that suggest that PFAS may be involved with other types of liver disease, and that's where our group has been studying, which has to do with fatty liver disease. This figure on the right is just kind of showing how the different ways that we can assess liver injury. So, for example, PFAS and PFOA, and there's indications for PFH excess, they cause damage to the liver. The liver releases an enzyme, a protein called alanine aminotransferase. That's just one of them. And that alanine aminotransferase, the doctor, when they give a blood draw, can measure it and measure its activity, and it's indirectly related to liver injury. And so this is one of the markers that we know is typically associated with PFAS and PFOA exposure. So in our laboratory, we're specifically interested in fatty liver disease. And I'm sure some of you have heard about it before, but fatty liver disease is where there's accumulation of fat in the liver. And not only is it not good for the liver's sake, because that liver can progressively get worse and get more inflamed and progress onto non-alcoholic steatohepatitis and potentially cirrhosis, but fatty liver disease is also associated with cardiovascular disease. So the liver makes fats, and then those fats kind of can get exported back into the blood as some of those like LDL types of lipids that you would talk about with your clinician. We also know that there's some indications that some PFAS are associated with elevation in markers of this type of liver disease. This is very recent work, and there's some indications that there's positive associations with some PFAS. And lastly, we do know that in rodent models, one specific, very obvious and clear association is that when rodents are treated with higher concentrations of just not PFAS and PFOA, but also PFH excess and some of the other replacements such as PFNA, PFDA, that is associated with fat accumulation in the liver. And so our group is interested in studying, is trying to understand whether or not those emerging PFAS are also associated with changing the liver behavior such that it increases fat accumulation within the liver. So I just want to point out just something, this is some collaborative work that we did with one of our other groups within our Superfund Center to kind of give you the wide breadth of where PFAS are detected. So this is an example of PFAS that were detected in the livers of pilot whale. And actually our Faroese cohort, one of the traditions that they do is eat pilot whale. So they get exposed in part to PFAS through this kind of traditional consumption. But what I want to point out to you here is that the very first column for all of these is liver accumulation. And so here we have PFAS. And for example, to give you perspective, so obviously PFAS highly accumulated in pilot whale liver, just as we see it in the livers of other types of marine life and human liver as well, for that matter. But also drawing your attention to the fact that there are all of these replacement PFAS that are not being regulated, that are still highly accumulating in these in whale liver. And our group doesn't see this just for whale, it's also observed for birds as well. Sometimes the profiles are not the same depending on where they're coming from, but the notion is that the liver is clearly a sink for many PFAS that are still being used in the commercial market, and some of those being used, for example, in AFFF materials such as PFHXS. So how do we study, and what are the challenges to studying PFAS health effects? Well, we know that there are about 7,000 PFAS on the commercial market, and we know that some of them are in mixtures in different types of chemicals for industrial use and commercial use, such as AFFF. There's no way that we can possibly target doing all of these in rodents. There's just too many. So we have to create in vitro models that are informative, relevant, and that can be used to study PFAS. In addition to the fact, sometimes we don't have a lot of this material to study, and so we don't even have really enough to be able to put into a large rodent study and get information about it. So our laboratory is able to screen those PFAS by using 96 well plate assays, in which we get many replicates, and it can be done rather quickly, and it's a starting point to understand the behavior of these replacement and alternative PFAS that are still being used. So our hypothesis for this work was that PFAS replacements can behave like some of our legacy PFAS, such as PFOA and PFAS in human hepatocytes. And that not only that, but these replacements may also possess these pro-fat accumulation types of properties in human cells. And so our hypothesis is that when we treat these liver cells with PFAS replacements, that these liver cells will also show characteristics that are associated with increased lipid deposition. So this is just generally, for those of you who are non-scientists, how it works. Human liver, so human donors, we get donor livers through a few different companies and academic sources. They isolate those hepatocytes in the laboratory, and the different parts of liver cells are kind of parsed out. And then those cells can go on dishes. They can be small or big, small or big, and they are incubated with culture medium and treated with different PFAS. And we can use that to understand behaviors that are related to how the liver would behave. In general, human cultured hepatocytes retain a lot of their important mechanisms that are very similar to how the human liver responds in situ, meaning that how the liver would respond in the body. And so we think that this is a solid tool to use for us to understand how the types of pathways, the type of signaling mechanisms that the human liver would also be responsive to. There are caveats to cell assays, but in general, we think that this is a reasonable tool to bridge understanding chemical and how the human liver will respond. So what we do is we actually take hepatocytes from about 5 donors. They are pooled. We have a large pool of this, so that means we can screen a lot, and it's kind of coming from the same source. They're plated onto 96 well plates. We treat them with the PFAS for either 48 hours and look at the change in their gene expression, so what genes go up and down, or we look at the ability of those cells to accumulate lipids within them. And then we compare those results to kind of our legacy PFAS. And what I'll note here is that the cells are treated with 3 different concentrations. So they go from what's 0.25 micromolar, so that's very low and an exposure relevant concentration, to higher concentrations that are much higher, which is 25 micromolar. So we do a dose response to kind of give us an idea about how potent the different PFAS are relative to each other. So I'm going to show you these heat maps. On the left are the different genes that we measure. We measure about 30 different target genes that are associated with different pathways, whether it's lipid metabolism, lipid transport, lipid synthesis and storage, cholesterol metabolism, and then things related to oxidative stress and also drug metabolism. And what I want to point out here is as the map gets more red, it means that the cells have responded more to upregulate some of those pathways. So when we look at this, we say, for example, see that PFOA and PFAS have some changes at very high concentrations, and so does PFHXS. And what I want you to notice as we go through the data, that as we look at lower concentrations, the question is, which PFAS tend to also induce these pathways more vigorously, but at lower concentrations? And how do they compare to this map? So here, just noting that PFHXS does behave very similarly to PFOA and PFAS. Now going on, these are some of our acid PFAS. And again, what we can see visually and very clearly is that there's a lot more red. And now we're starting to see this upregulation of gene expression at much, much lower doses. You know, this is really an exposure relevant concentration. So this is a concern because they are turning on pathways and doing it in such a way that's very similar to PFAS that we know have been banned, or excuse me, voluntarily removed. So we also see some of this behavior with some of our long chain PFAS, but not to the same degree. And if you can see, the pattern does kind of look different, and it tends to only occur at very high concentrations, except for the C11. When we look at other PFAS replacements, such as basically this is a Gen X or 6,2-FTS, we also see a similar trend, especially for 6,2-FTS, that looks very similar compared to our legacy PFAS and at lower concentrations. And what I would say here is that this particular component, we're also, our group's also seeing it in bird livers in high concentrations. So we're currently screening human livers from human donors to see if we can also see this particular PFAS in them as well. In addition to this, we also have what's called FOSAs. These eventually get broken down into PFAS, and these also show, again, a similar profile, and again, at lower concentrations. So I think our concern as researchers here is that not only do these behave like PFAS, but they also tend to, at least in our models, be more potent. So what does that mean? So the next thing we did was we asked, okay, this is gene expression, but how does this translate into how the cells behave? And so we tested lipid accumulation in the cells. So again, in non-alcoholic fatty liver disease, the liver gets fatty. And the way the liver gets fatty is either that it's taking up more free fatty acids from the blood and then converting those free fatty acids to a storage lipid called a triglyceride, or they're taking glucose and they're going to go ahead and convert it within the cell to triglycerides. And within that disease state, both of those processes occur. And so in our model, we're not adding free fatty acids, but we're basically looking at this pathway. Does this pathway get turned on where cells are now able to convert the glucose that's in the media to lipids? So first we looked at our legacy PFAS, and they don't really induce much lipid accumulation within the cell. So what you're looking at here, the blue represent the nuclei of the cells. The red represent staining of lipids within the cell. So if you see red, that means the cells have more lipids. This is a positive control. So we know when the cells are treated with palmitate and oleic acid, there's a lot of lipid accumulation in the cells. So here we can see with whether it's PFOA or PFHXS or PFAS, there's really no difference with their red staining compared to the vehicle control, which is dimethyl sulfoxide. And here, this is a graph just demonstrating that. So we can see that there's a large change in lipid staining associated with our positive control. But then when we did our different treatments of PFOA, PFHXS, and PFAS, there's really no difference than our vehicle controls. On all of the graphs, the vehicle controls are just kind of here, this dashed line. So here, when we look at our short chain PFAS, this should say PFAS, excuse me, not PFAS, but our short chain PFAS, just like the gene expression profiles that we saw, they also were able to induce lipid staining within the hepatocytes. And this is quantified here. It's not tremendous, but again, what we're doing is we're forcing the cells to make lipids. And so if we gave them more materials to do so, which we plan to do, then we would potentially see a greater response. But this is one of the first studies that actually shows that PFAS can do that. Long chain PFAS also can induce, some of the long chain PFAS can also induce lipid accumulation. So again, here's our positive control. When we looked, there were some of these longer chain PFAS that did, and we can see that there's also more red staining within those cells. Of additional PFAS, replacements do. So again, we mentioned that 6,2-FTS induced a pretty strong signature for the gene expression. And here we can also see, here's our controls. So these are ones that just received vehicle. Here's the ones that are our positive control. So these just show that the assay worked, and you can see the little lipid droplets within the cells. And then here, for example, is 6,2-FTS. And so 6,2-FTS increased the amount of lipid staining within these hepatocytes. And then the graphs kind of display that as well. GenX did not. So these alternative PFAS, in addition, such as we also looked at FOSA, MET-FOSA, and ET-FOSA, and compared to our DMSO controls, these also induced an increase in lipid staining. And if you recall, this matches pretty well with our gene expression data that also showed that they were turning on this hot signature that was associated with gene expression for lipid accumulation. So in summary, it appears that replacement PFAS and PFAS alternatives can induce gene expression that's similar to our legacy PFAS. However, these replacement PFAS and PFAS alternatives seem to be more potent in our system, and we can have a discussion as to why we think that's the case. We do know that these alternatives can also induce lipid accumulation in human hepatocytes, and that typically the shorter chain PFAS were often more potent. Along with that, I would say that 6,2-FTS was also, and the FOSAs were also potent. And lastly, PFAS, we would conclude from this that our PFAS have properties in our cell system that suggest that they can induce fat accumulation in human liver cells. And so because of this, these properties really do need to be studied more in human epidemiological studies. There's a recent publication from early 2021 that showed an association in children between a few, such for example, PFHXS and fatty liver disease in children. So I think that these cell-based studies make an argument that we should go forward and study more of these in rodent and very much so in human studies. So with that, I would like to thank the organizers. I would like to thank Jeff Burgess and the Miami crew as collaborators. Certainly acknowledge NIEHS and our Harvard STEEP Superfund Center, as well as Dr. Emily Marks, whose thesis work this was. She was supported by her other fellow student, Marissa Foll, and my laboratory technician, Wee Wee. So thank you for your attention. I hope I didn't go over, and thank you to the inviters.
We're just going to let you do Q&A via Zoom.
We were having some technical difficulties, but I'll facilitate our Q&A session. So first of all, thank you for an amazing presentation. It's always amazing to listen and hear a lot of the basic science and toxicity that goes into PFAS. We have some questions from our audience, both in the room and online. So I'll be asking you a couple of those questions to facilitate them over to you. So could you please elaborate on the differences between C8 or C6 PFOA and the alternatives to PFAS? Will these alternative PFAS end up in our firefighter gear?
Oh, I don't think I can answer whether or not those could end up in your gear. So, however, I think what we are seeing is that these replacements are being used more and more. And our grant is in year 5. And what I can tell you is when we first wrote the grant, there were about 2,000 PFAS out in the kind of that were either being detected or had been kind of registered for commercial use. And it's just grown, it's beyond exponential at this point. So I think that the concern will be that I think we will have to be concerned that those types of chemicals can get into, fabrics, gear, you name it, and we're always going to be chasing. So I think it's possible. I don't know if they will. They certainly have they do have a lot of similar properties. I think our concern is that they behave a lot like the ones that are already out there. We're not seeing a lot that are behaving differently. And I think the field has to think about how to engineer these a little differently, with a lot of use of understanding their biological pathways and systems and how they get into biological systems.
Thank you. For the audience, we have a very different heterogeneous audience with us, including our firefighters, government, even international members. And one of the questions that we're seeing from our audience is how does different dosages of exposure within animal models might differ in the exposures that are seen, for example, in human models, so that you can interpret the health effects from them?
Okay, that's a really good question. So in general, when you saw my data, for example, we used the lowest concentration we used was 250 nanomolar. And that's a reasonable concentration compared to what we know is detectable in liver autopsy tissue from humans. Now, as PFAS and PFOAs, as they have been withdrawn for certain uses, we know that those concentrations in the human liver have gone down. But in general, we try to put our lowest concentrations around what we would see potentially in a human liver cell, like a human autopsy and necropsy tissue. Once we get up to 2.5 micromolar and 25 micromolar, those are way, way high. And we're really just trying to push the cells to see if they can respond at all, right? So if we give them a really high concentration and the cells don't really do much, then we know that we can kind of interpret that might be like a less sensitive model. It's kind of complicated, but what I would say is that as that lower concentration is a bit concerning, because it means that they can get in. I didn't present it today, but we actually did a lot of treatment on our hepatocytes with concentrations that we would find in well water, so parts per trillion concentrations, and the cells do respond to that. So that's a bit of a concern that we can go that low. I think there's limitations to cell models that we have to be very careful about totally addressing potency. I think it's like if you look at the cell models across, the PFAS across one model, you can understand the differences. But we have to be careful about drawing absolute conclusions based on concentration. So what I would say is low concentrations stimulate cells to do something, and that's probably the concern. Because these still have relatively long half-lives compared to drugs, like I come from a school of pharmacy, we do have to be concerned. Some things like PFBS, we're starting to see more appear in human serum than we were seeing. And so now we have to start thinking about, are people getting exposed to more? What does that mean? Yes, these do get to the liver. They do turn on gene expression. So it's very complicated.
Indeed. We're thankful you're at the home sort of addressing some of these questions. We have more from our online audience, and the question is, in your study, were any of the donor livers of those of a firefighter? And if so, were the findings different from the general population? I guess the donor base for the livers.
Yeah, so I don't know that information, so I can't really tell you that, but I would probably say no. So in this case, because we wanted to screen so many with so many different assays, these came from the same five donors. And we just know that there were 3 males and 2 females put to that donor population, and that these were considered, quote, normal. These livers would have been kind of diagnosed as normal, or the and the hepatocytes that were isolated at that time were considered kind of normal, healthy hepatocytes.
Yeah, that makes perfect sense. In terms of sort of immune function, could you describe for the layperson, like how does PFAS sort of interact to impact immune function in animal models or in human models?
Yeah, so that's a very complicated question. So in our models, we did not co-culture with immune cells. The liver has immune cells. We did not specifically do that. Now, there are studies to show that PFAS, so for PFAS, PFOA, some of the other PFAS can be associated with decreased response to vaccination. That's one type of immune response. What's not clear is there's also called the acute phase response. So that is your response to infection. And so in those, it's a little less clear about how PFAS in humans is associated with that immune response, because there's some negative associations, meaning that the higher the PFAS, the lower that particular immune response marker. But in general, I think the most consistent data has to do with PFAS exposure and decreased to an immune stimulus that would be associated with vaccination. So decreased response to the children had gotten their MMR vaccines and they had a decreased response to that. And there's some evidence that there's a decreased response to infection in children that's measured by fever and other responses to infection. And that's one aspect of the immune response.
And our last question today is actually about detoxification. So is there any effective mechanism that you're aware of for detoxing the liver when there is exposures to, say, PFAS or PFOA? I feel like you're getting these big, broad questions that the group wants to know, but
Sure.
Thank you.
Yeah. So here's this is something kind of being very actively studied. And so there's some thoughts that blood donation can help, because obviously if you're taking that away, your body is now synthesizing new proteins. So one of the thoughts, I don't know how effective it is blood donation. And I think that comes from understanding that when women are menstruating, that their levels can get lower. So I think that's the rationale behind it. We're actually interested in studying ways to prevent absorption so that the person would then be eliminating more. So we do have some studies in rodents to suggest that if you can block certain mechanisms of gut absorption that kind of are similar to drug absorption, that we may be able to enhance elimination. But clinically, there's not a lot of options. There have been some studies around cholestyramine. Alan Duchteman published a paper where they gave some bile acid sequestrants, and those were associated with a kind of enhanced elimination. So I think that this is something we do really need to work on. I know there's a group out of Kentucky that was looking at the lipid proteins in your blood and exercise. And so if you're maybe changing the profile of your cholesterol and cholesterol, like LDL, VLDL, things like that, if you're changing those profiles, because they can maybe carry PFAS, that they may also change that elimination. But I would say right now that's a challenge for the field, and we're trying to figure it out. Last point on this, there's some evidence that in the environment, they're using something called cyclodextran to sequester it. And cyclodextran, from a molecular standpoint, kind of looks like a tube. And the thoughts are, because cyclodextran is in some drugs as fillers, that maybe actually cyclodextran could be a potential sequestrant that would help you sequester some of that you're absorbing, and maybe that would help with elimination. But that hasn't been tested, but interesting, it's being tested in the environment. So it could be tested in humans.
So many promising opportunities for the future. Thank you. And so we're just at time. And so on behalf of our planning committee and our audience, both international and here local in Orlando, thank you so much for that amazing presentation. So for everyone in the room, we're going to go, and online, we're going to take a 5-minute break. We'll resume back at 11:05 a.m. So thank you.